The protein kinase CK2 is involved in a wide range of biological processes and cellular functions, including inflammatory responses and pathologies associated with inflammation. Also, its aberrant expression and activity are characteristic of many cancers. Yet the basic function of CK2 in CD8 plus immune T cells has remained completely unknown. CD8 plus T cells, also known as killer T cells, are critical to maintain health by controlling infections by intracellular pathogens, including viruses and intracellular bacteria. CD8 plus T cells also can control cancer progression by directly killing cancer cells, and through recruiting and activating other immune cells. Now, in a study published in the Journal of Immunology, University of Alabama at Birmingham, researchers report that protein kinase CK2 controls CD8 plus T cell effector and memory functions during infection. These experiments were done in mouse CD8 plus T cells and a mouse model of infection by the intracellular pathogenic bacteria Listeria monocytogenes. The CK2 kinase is known to act in cellular signaling pathways by phosphorylating target proteins at specific serine or threonine amino acids. The CK2 kinase has 2 catalytic subunits and 2 regulatory subunits. The UAB researchers, led by Hongwei Qin, Ph.D., and Eti Tika Benvenisti, Ph.D., deleted the CK2 alpha catalytic subunit from mouse CD8 plus T cells and compared them to CD8 plus T cells with an intact CK2 kinase. The researchers first showed that the CK2 subunits were induced, and that CK2 kinase activity was increased, when intact CD8 plus T cells were activated by stimulating the T cell receptor to trigger an immune response. That showed a potential role of CK2 in CD8 plus T cell responses. In contrast, the CK2 alpha deletion mutants showed significantly less CD8 plus T cell activation and proliferation, indicating that the CK2 alpha activity was required for those steps. Furthermore, the researchers say their findings suggest that CK2-alpha is involved in CD8 plus T-cell metabolic reprogramming during activation, specifically through increases in glycolytic metabolism and mitochondrial respiration. Mitochondria are the powerhouses of cells, supplying most of the cellular energy. Upon activation, intact T cells are known to undergo substantial metabolic reprogramming, but the UAB researchers found that CK2-alpha deletion mutants had significantly less glucose uptake and metabolism, and decreased mitochondrial respiration, compared to T cells with normal CK2. Mechanistically, the UAB team showed that CK2-alpha controlled the reprogramming of CD8 plus T-cell metabolism by regulating the well-known AKT-mTOR signaling pathway. Using a mouse Listeria monocytogenes infection model, Qin, Benvenisti and colleagues found that CK2-alpha was required for CD8 plus T-cell expansion, maintenance and effector function in both primary and memory stages during infection by the intracellular bacterial pathogen. Taken together, our study demonstrates that CK2-alpha regulates CD8 plus T-cell activation and differentiation both in vitro and in vivo, Benvenisti said. However, the biological function of CK2-alpha in CD8 plus T cells, particularly CD8 plus T cell cytotoxic function, remains to be explored. Co-authors with Benvenisti and Qin in the study, protein kinase CK2 controls CD8 plus T cell effector and memory function during infection, are Wei Yang, Hairong Wei, will make a difference. Sponsored Opportunity StorySource, materials provided by University of Alabama at Birmingham. Original written by Jeff Hansen. Note: content may be edited for style and length. Journal reference: Wei Yang, Hairong Wei, Gloria A. Benavides, William J. Turbot, Jessica A. Buckley, Xiaoshanu Yang, Liana Zhou, Jianhua Zhang, Lori E. Harrington, Victor M. Darley Usmar, Hongwei Chin, Etienne Benvenisti. Protein kinase CK2 controls CD8 T cell effector and memory function during infection. The Journal of Immunology, 2022, 209, 5, 896 doi, 10. 4049, Gimmunol. 2,101,080. Cite this page, University of Alabama at Birmingham. Protein kinase CK2 has key role in killer T cells during infection by Listeria monocytogenes. Science Daily. Science Daily, 1 November 2022. ift.tt/gkbdp. University of Alabama at Birmingham. 2022, November 1. Protein kinase CK2 has key role in killer T cells during infection by Listeria monocytogenes. Science Daily. Retrieved November 1, 2022 from ift.tt/gkbdp. University of Alabama at Birmingham. Protein kinase CK2 has key role in killer T cells during infection by Listeria monocytogenes. Science Daily. https://ift.tt/gkeabdp, accessed November 1, 2022. Thanks for watching. Please subscribe my channel for more videos.